Okay, so the enzymes involved in drug metabolism in general are present in many tissues, but by far they are more concentrated and more frequent in the liver. So the liver is the main site of drug metabolism. And for orally administered drugs, this is very important because what it means is that the drug can be metabolized um, in the GI tract first and then immediately by the liver because the GI tract is connected anatomically to the liver via the portal vein. And so this is called the first pass metabolism. And because of this effect, many drugs can actually not be administered orally because they get completely metabolized already in the first stage. Let's look a little bit about at the two types of enzymes we already discussed. And so we have phase one enzymes which carry out reactions on the molecule itself. Those will be cytochrome P450 is going to be the main family of enzymes we look at. Another very important family is given by the monoamine oxidases, which metabolize neurotransmitters. Phase 2 enzymes are typically named after the molecule they transfer, for example, N-acetyl or acyl coenzyme A, and then they have at the end a transferase, indicating that you know their job is to transfer that group to whatever functional group in the drug we have. Okay, let's look a little bit of at the mechanism of actions for these drugs. And so we have different possibilities. We can have ultimately all this mechanism end up with the same product, but we are starting from a molecule that contains a carbon skeleton and a heteroatom of some type with a lone pair on it. And we can either have a single electron transfer reaction leading to radical intermediate and then to this adduct. We can have a hydrogen atom transfer, so the transfer of a atom of hydrogen with its single electron to form a radical intermediate once again, and then evolves to this intermediate. Or we can have a hydride transfer to yield directly this intermediate. And different enzymes will utilize different mechanisms, but as I said, ended up with the same type of reaction. So let's look at practical views of this. So the mechanism that we have seen, so this, particularly this intermediate is the same for all these reactions. So we always have the same type of positively charged heteroatom here. If we start from an alcohol, we'll end up with an aldehyde. If we start from an ether, we'll end up with aldehyde and alcohol. So we'll have actually breakage of the oxygen to our group here, our prime rather. For amines, this reaction will generate aldehyde and an amine. So it's what we called an oxidative deamination. And for halogens also we'll end up with an aldehyde and acid. Most of these reactions are carried out by cytochrome C450, which is basically the most important enzyme superfamily. Those are heme proteins, and there are more than 50 families, each of them with up to 10 superfamilies, subfamilies. These enzymes are found mainly in the liver, but also in small intestine, kidney, lungs, brain, and other tissues. They tend to be associated with the membrane in the ER, so the endoplasmatic reticulum, and a portion of them containing the active site and the pocket for the substrate recognition is exposed to the cytosol. We have luckily the crystal structure of many of these enzymes, so where we can see that again they contain both a heme. In this case, this is one of these enzymes that has coordinated warfarin, which is a anticoagulant drug. There are 5 main subfamilies of these enzymes, and they basically are different by sequence, but generally speaking, their main, the active site and the mechanism of action is the same. They're very versatile enzymes, and they tend to be not too specific in the substrate they recognize. So let's look a little bit more in detail at the mechanism of action of these enzymes. As we already indicated, they all have a heme at the active site. And so the resting state is this one, the iron 3 rather state. We have a reduction. Well, actually, in the first step, we have coordination of a drug to form this enzyme drug adduct. Ultimately, all these enzymes do the same thing. They transfer oxygen to a substrate. And they do that by utilizing molecular oxygen and NADPH through an enzyme, which is a flavoprotein called cytochrome P450 reductase, which is a source of electrons and powers the whole metabolism here. So in the second step, once we have formed this adduct with the drug, we have a reduction by 1 electron to form now the iron 2 state. And the iron 2 or ferrous state of the enzyme is the one that can go on to coordinate molecular oxygen. At the same time, this adduct gets reduced to form this complex, which is also called compound 1, which is where the whole chemistry happens. And so in the next step, we already had this reduction again from the same flavoprotein that reduced, donated the first electron. And the fourth step is the one where we have the whole chemistry. This activated oxygen, one of the atoms is transferred to the drug to form this hydroxy group. The other atom of oxygen forms water, so we have concomitant uptake of two protons. And then, the drug is released is a metabolized form, and cytochrome P450 is released in its resting state, which is the same as the initial one. Let's have a quick look now at what the heme groups look like. Is iron protoporphyrin 9, and so at the center of it contains an iron, as I said, in the resting state. That will be an iron 3. And let's look at how compound 1, so the active species, is generated. This is the active species, and in particular this iron 4 adduct. So this is just another look at what we just went through. And so we have reduction of the iron 3 to iron 2, coordination of oxygen to form this intermediate, and that in turn gets further reduced to form now transfer another electron to form now the activated oxygen here, an iron 3. And then we have loss of water, so breakage of this bond, and formation of compound 1, which is highly reactive. How does this compound 1 react? Well, it is thought that will take up a proton, or rather, a radical, an atom of hydrogen. As you remember from organic chemistry, this arrow with a single barb indicates that only one electron moves away, and so we form this hydroxy adduct and a radical on the substrate. So the radical is very reactive and ends up uptaking the hydroxy group, and again we have had homolytic breakage of the bond with one electron going to the iron and one electron going to the oxygen here. And so we generate the hydroxylate form of the substrate and repristinate the normal iron 3 resting state. This is what happens, for example, for nicotine, which gets hydroxylated at this position by cytochrome P type of enzymes. And it's a very common reaction. In fact, hydroxylation reactions are some of the most common metabolic reactions encountered by several substrates, and here's a list of possible functional groups, so possible substrates, and what the product ends up being upon hydroxylation. In the next video, we will look at the specific mechanism for some of these reactions.